Hello students. Today we'll see how plasma is prepared from blood. Let's start with the introduction. Blood plasma is the liquid component of blood in which blood cells are suspended. It makes up about 60% of the total blood volume. It is composed of mostly water, which is 90% by volume, and contains dissolved proteins, glucose, clotting factors, mineral ions, hormones, and carbon dioxide. Plasma being the main medium for excretory product transportation. To minimize the biochemical changes that take place in vitro, blood which is collected must be stored in a refrigerator at 4 degrees Celsius. Plasma is the supernatant fluid obtained when anticoagulated blood has been centrifuged. The blood is mixed with an appropriate amount of anticoagulant like heparin, oxalate, or ethylene diamine tetraacetic acid, EDTA. Anticoagulants are the agents or chemicals which prevent the coagulation of blood. Blood tends to clot immediately after withdrawal from blood vessel. Anticoagulants are used to prevent clotting or coagulation of blood for further use. EDTA is generally recommended for morphological studies of blood like RBC, WBC, thrombocyte count, differential WBC count, hemoglobin estimation, etc. 1 to 2 mg of EDTA is a recommended quantity per ml of blood. This preparation should be mixed immediately and thoroughly to avoid clotting. Blood serum is blood plasma without fibrinogen or the other clotting factors. Serum is clearer than plasma because of fewer proteins. Proteins are sometimes considered as interfering substances in some tests as they react with the reagent and thereby yield inaccurate results. Serum is the preferred specimen in clinical testing as interference that may be caused by a plasma specimen because of the presence of an anticoagulant is eliminated. Process the samples as soon as possible. If storage is necessary prior to processing, store the blood at room temperature, shielded from light and on a rocker. And do not refrigerate the cells. Now let's look at the blood plasma preparation. Materials and equipment required. Human blood sample, vacuum tubes containing anticoagulant, serological pipettes of appropriate volumes which are sterile, centrifuge tubes, cryovials, and a benchtop centrifuge. Let's look at the procedure. Draw blood into vacuum tubes containing 1.8 mg EDTA dipotassium salt per ml blood. Be sure to draw the full volume to ensure the correct blood to anticoagulant ratio. Second, invert the vacuum tubes carefully 10 times to mix the blood and anticoagulant and store at room temperature until centrifugation. Samples should undergo centrifugation immediately. This should be carried out for a minimum of 10 minutes at 1500 rpm at room temperature. Do not use brake to stop the centrifuge. Number 4, this will give 3 layers. From the top to bottom, you have the plasma layer, the leukocytes layer as a buffy coat, and the erythrocytes layer. Carefully aspirate the supernatant, which is the plasma, at room temperature and pull in a centrifuge tube. We should take care not to disrupt the cell layer or transfer any cells. Inspect the plasma for turbidity. Turbid samples should be centrifuged again and aspirated again to remove the remaining insoluble matter. A general recommendation is cold handling, which is 4 to 8 degrees Celsius of plasma. It should be stored at minus 80 degrees Celsius or lower for long-term storage. Aliquot plasma into cryovials and store at minus 80 degrees Celsius. Ensure that the cryovials are adequately labeled with the relevant information, including details of additives present in the blood. So, this is how plasma is separated from blood. The prepared plasma can be used for further analysis. It must be noted that serum and plasma specimens are of better quality for analysis if smaller volume aliquots are prepared before being stored for later use. Standard blood sample collection and preparation methodologies should be followed if the research testing results are to be translated to future clinical and diagnostic tests, like the type of additive used to blood collection tubes, sample processing time or temperature, hemolysis of sample, and sample storage parameter, and the number of freeze and thaw cycles. These types of differences between samples can affect the outcome of the result of the analysis. Hello students, today's experiment is on differential staining of WBC. Let's go through the introduction first. The cellular components of blood include erythrocytes or RBCs, leukocytes or WBCs, and platelets. Normal human blood contains 4.8 to 5.2 million erythrocytes per ml and 4,000 to 10,000 leukocytes per ml. Leukocytes or white blood cells, WBCs, can be broadly divided into granulocytes and agranulocytes. Granulocytes are classified as neutrophils, eosinophils, or basophils on the basis of cellular morphology and cytoplasmic staining characteristics. The neutrophil has a multi-lobed nucleus and a granulated cytoplasm that stains with both acid and basic dyes. It is often called a polymorphonuclear leukocyte, PMN, for its multi-lobed nucleus. Neutrophils are the most abundant white blood cell, constituting about 60 to 70% of circulating leukocytes. They are phagocytic and defend the body against bacterial or fungal infection. They are usually first responders to microbial infection, and their activity and death in large numbers form pus. The eosinophil has a bilobed nucleus and a granulated cytoplasm that stains with the acid dye eosin red, and hence its name. Eosinophils constitute about 1% to 3% of circulating leukocytes, and like neutrophils are motile phagocytic cells that can migrate from the blood into the tissue spaces. Their phagocytic role is significantly less important than that of neutrophils, and it is thought that they play a role in the defense against parasitic organisms. The secreted contents of eosinophilic granules may damage the parasite membrane. Basophils are non-phagocytic granulocytes that constitute less than 1% of the circulating leukocytes. The basophil has a lobed nucleus and a heavily granulated cytoplasm that stains with the basic dye methylene blue. They function by releasing pharmacologically active substances from their cytoplasmic granules. These substances play a major role in certain allergic responses. Agranulocytes are categorized into monocytes and lymphocytes. They are also known as mononuclear cells. Monocytes are the largest type of WBCs, phagocytic like neutrophils, so they are also called mononuclear phagocytes. Monocytes eventually leave the bloodstream and become tissue macrophages, which remove dead cell debris as well as attack microorganisms. Lymphocytes constitute 20% to 40% of the body's white blood cells and 99% of the cells in the lymph. Lymphocytes are distinguished by having a large, deeply staining nucleus and a relatively small amount of cytoplasm. The lymphocytes can be broadly subdivided into two populations, B cells and T cells. Now let's come to the objective of this experiment. The objective of this experiment is to differentiate the WBCs or white blood cells or leukocytes using different stains based on their morphology and staining characteristics. The principle of this experiment is the stained WBCs can be differentiated into many types depending on their physical cell characteristics like size and shape of the cell, size, shape and location of the nucleus, position, size, shape and color of the cytoplasmic granules, and the appearance of the cytoplasm. The materials required for this experiment are: number 1, microscope; 2, clean glass slides; 3, staining rack; 4, cedarwood oil; 5, distilled water; 6, staining solution, which can be either Leishman stain, Wright stain or Giemsa stain; and 7, blood sample. Leishman stain. The composition of Leishman stain is Leishman powder, which is made up of methylene blue and eosin, 0.15 gram in 100 ml of methanol. Wright stain. Wright's powder, which is 0.1 gram in 60 ml of methanol. And for Giemsa stain, the composition is 3.8 gram of Giemsa powder, 200 ml of glycerin, and 312 ml of methanol. Dilute stock stain of Giemsa stain 10 times to make the working stain. Now let's come to the method. Preparation of blood smear. Place a small drop of fresh or anticoagulant mixed blood on a clean, grease-free glass slide about 2 cm away from one end. Next, place another slide, that is the spreader slide, in front of the blood drop at an angle of 45 degrees to the first slide. Then, draw the spreader slide backwards till it touches the drop of blood. Let the blood spread across the surface between the two slides. And then, with a single swift forward movement of the spreader slide, spread the blood along the surface of the slide. Now dry the smear immediately by waving the slide rapidly or exposing it to air. The rapid drying of the smear prevents granulation of the cells. Now, staining of the blood smear with Leishman stain or Wright stain. Place the slide on a staining rack, then cover the entire blood smear with Leishman or Wright stain. Add equal quantity of distilled water and thoroughly mix it with the stain by rocking the slide. Let it stand for 5 to 10 minutes and then wash off the stain gently. Air dry the smear and then examine it under microscope using high power objective. Now let's see how the blood smear is stained using Giemsa stain. Place the slide on a staining rack and then add 5 drops of methanol on the blood smear to fix the smear. Wash the slide with distilled water after 2 minutes. Now place the slide on the staining rack again and cover the smear with Giemsa stain and leave it on for about 45 minutes. Do not allow the stain to dry up during this period. Then wash off the stain with distilled water. Air dry the slide and examine it under the microscope. Let's come to the result of the experiment. When observed under the microscope, the different types of WBCs or leukocytes appear as follows. Neutrophils. They are about 10 to 15 microns in size. The nucleus has 3 to 7 lobes, and the cytoplasmic granules are dust-like, light, violet or pinkish in color. Eosinophils, they are 12 to 15 microns in size, bilobed with round and refractile cytoplasmic granules which stain red or orange with acid dyes. Basophils, they are 11 to 14 microns in size, bilobed or trilobed nucleus with deep blue cytoplasmic granules of various sizes that take up the basic stain. Lymphocytes are of 8 to 15 microns in size. The nucleus is round and occupies most part of the cell. There is little cytoplasm which possess no granules. Monocytes are 16 to 18 microns in size. The nucleus is convoluted, kidney or horseshoe shaped. No granules are observed in the cytoplasm. Now let's come to the conclusion. This experiment demonstrates how to stain and differentiate WBCs using different dyes. Neutrophilic granules stain light violet or pink since they are weakly stained because of their neutral nature. Eosinophilic granules contain a spermine derivative which is alkaline and thus stain strongly with the acidic component of the dye, that is eosin Y, giving a red or orange color to the granules. Basophilic granules contain heparin, which is acidic and takes up the basic component of the dye, methylene blue, and thus stains deep blue.